Hello again. I mentioned in earlier that a bacteria has genes that can be used to metabolize different forms of sugar. So it has genes to metabolize glucose, it has genes to metabolize lactose, and so on. When glucose is absent, right? When glucose is not present, then this will be the time that it looks for other sources of sugar in order for it to get carbon. So in this case, an example of an operon that we are going to study is the LacOperon. It's a lactose operon. Now, lactose operon, it metabolizes lactose, and it has an inducible system with, so here you go, it says it's negatively controlled inducible operon. Well, you know it's not inducible. Inducible means normally it's off, you need to switch it on. Negatively controlled means you need to take out the repressor to switch it on. Right? If you don't remember this, you just need to go through the slides again, and then it's not really that complicated. It's just that you just need to get used to it. So it's negatively controlled inducible operon. Now, okay, the genes are only expressed in the presence of lactose. So this is the structure. This is a lactose operon, right? The lac gene, and within that LacOperon, you have three genes. You have gene Z, gene Y, and gene A. You have the beta-galactosidase, you have the galactoside permease, you have the transacetylase, and then you have the promoter and operator. And then this is the regulator gene that has the lac repressor, and then that's the promoter for the gene. So you have two different promoters. This promoter is for the gene I, the inhibitor or the repressor molecule. This promoter and operator is for the three genes. So this is called the whole operon. All right. Now, what does these genes do? You can read up, but basically, beta-galactosidase is the gene that's involved in metabolizing lactose and breaking it down into smaller molecules. I think this is just an information on the next slide. Here you go. You have your lactose, right? Under the influence of beta-galactoside, there are two pathways. One pathway is the breakdown of lactose to glucose and galactose, or the beta-galactosidase can also be used to produce allolactose. Now here, allolactose is the inducer. Where have you seen that? You've seen that in examples of the how operon functions. Now, this is this slide is a bit complicated, but never mind. So I won't be explaining that much of this, and I won't be asking you too much on this or too much of this in the exams. All right. Okay, but basically how it works, you have the allolactose as an inducer. Remember this name, allolactose. You have the lac repressor. So let's just use this and then see if it applies here. Right? So what you need, so you can imagine if that is the allolactose, this is the lac repressor molecule, right? It binds. With the presence of the inducer, the allolactose will bind to the lac repressor and then changes its shape, and then resulting it to not be able to bind anymore, and because of that, polymerase can start transcribing. All right. Okay. For this side, I want to talk about catabolite repression. What is catabolite repression? Now, I mentioned that bacteria prefers glucose because it's the simplest sugar. I also mentioned that the lactose operon will be switched on in the presence of lactose. Correct? Right? So what happens if you have both glucose and lactose? Right? Do both operons get activated? Now, for this matter, we want to have a look at catabolite repression. Okay, I think we'll stop there for now, because I need to do another video just to look at this